There are faculty and laboratories that study the basic development of how individual kinds of neurons are born, develop, connect, and function. And that could be in the retina, in the inner ear, in the part of the brain that has to do with ALS, in the part of the brainstem that has to do with Parkinson's disease. And our feeling is, our approach is, that understanding how those cells develop is essentially identifying the recipes for building them. And ultimately, what we'd like to do is rebuild them. So we have a strategy of monthly interlab meetings where laboratories, not just within hearing or vision or motor systems, but across these groups, all get together on a Tuesday morning for an hour or two, share their latest data, discuss, argue, and raise new ideas that come, let's say, from Parkinson's disease work into the visual or hearing system, and vice versa. One goal is to build, through molecular recipes, billions of the right kinds of nerve cells, or glia, those that degenerate in individual nervous system diseases, like ALS, Parkinson's, retinal degenerations. And then screen, look, investigate for new therapeutics, for drugs, or for high-tech bioactive molecules. And we're exceptionally lucky to have, as a colleague and collaborator, the Therapeutic Screening Center directed by Professor Lee Rubin. And we're lucky for two reasons. One, the facility enables each of these subgroups to bring a collaborative project to screen for drugs or bioactive molecules for the cells that degenerate in ALS, the cells that degenerate in the childhood relative of ALS called spinal muscular atrophy, for those that degenerate in Parkinson's disease. But we're also incredibly lucky because Professor Rubin is not just a therapeutic screening world expert, but he's a world-class neurobiologist. So we don't just bring cells to a center and hope that somebody will look for drugs, but we collaborate with a colleague where we can think together about the best possible way to screen for drugs. And that would be nearly impossible were it not for HSCI and this bringing together of the facilities, the center, the infrastructure, and the investigators. The HSCI IPS, or Induced Pluripotent Stem Cell Core, has generated a series of what we term lines of IPS cells from patients with individual neurological neurodegenerative diseases, from ALS, from spinal muscular atrophy, from Parkinson's disease, from children with autism, from Alzheimer's disease. We can build those lines into iPS stem cells that have many similarities to ES cells, and then using the molecular recipes that we're identifying in our joint research, bring them back to Lee Rubin. So together, HSCI has built this community where the wherewithal to purify cells comes together with faculty that study those cells, to find the recipes, to make those cells, to bring them to Lee Rubin, and it's all connected. And it's that connectedness that is what makes HSCI exceptionally important.